I'll be starting off this presentation by briefly introducing our assignment. This project is part of our STT1083 Introduction to Plant Genetics course, and our main objective is to construct a phylogenetic tree using the software Mega12. A phylogenetic tree is a diagram that shows the evolutionary relationships between different DNA samples or taxa. By analyzing these relationships, we can determine which taxa are closely related, which ones have undergone the most genetic changes, and which ones serves as the outgroup. In this video, we'll walk you through the step-by-step process of sequence alignment, model selection, and tree construction, all using Mega12. Let's get started.
In sequence alignment and click on DNA. Select sequence 1 and proceed by click delete. Then insert sequence from file by select insert sequence from file, locate it and open data set 1 in MegaX. Then align the sequence using cluster W and confirm the choices and started the alignment process. After align the data, verify that no more sequence left to delete before we proceed. Finally, save the aligned data by navigate to data and save the file, saving it as dataset element 2025.mas. Complete the process by click save. Move to step 2, model, and select find DNA model to determine the optimal DNA substitute model. Then choose analyze preference required, then click OK to initiate analyze process. Monitor it to make sure the process successful. Step 3. Started by analyze all the model and ready to proceed the base model. The top selected model will be listed as optimal choices. Then click on icon phylogeny. Click construct or test maximum likelihood tree. Then select yes. In model method box, then choose base model identified early like Tamura tree parameter, then confirm and select OK. Review the selecting figure display. The phylogenetic tree that constructed based on aligned sequences.
Explaining the relation between taxa sp2, 5, sp2, 15, sp2, 6, sp2, 17, and sp2, 14. These taxa are grouped closely together in the same clade. They likely share a region, common ancestor, and are closely related. sp2, 16, it is connected to the clade above, but on a spread branch. This suggest is moderately related to the group. sp2, 11 and sp2, 1. These two taxa from a small clade. This indicates they are more closely related to each other than to the other taxa. sp2, 7, sp2, 13, and sp2, 12. This from a small group branching for a share not. They are evolution related but not as tightly as other clades. sp2, 8, sp2, 2, sp2, 4, sp2, 10. These taxa are somewhat related and from other intermediate group. These groups show moderate similarity, branching earlier than other type clades. sp2, 3, sp2, 9, sp2, 18, sp2, 19, and sp2, 2. These are grouped in the lower section of the tree. They are share a common evolution origin and are closely related with the group. sp220. It is branched out alone from the main group. This indicator is the mostly genetically distinct taxon in the tree. Taxa that are clustered together are most closely related. Longer branch lengths of isolation position suggest more genetic divergence. The three show how all the taxa share a common ancestor at the same point but evolve at different rates and direction.
The taxon sp220 show the longest branch length in the phylogenetic tree, indicating that it has undergone the most genetic change compared to the other taxa. This is because longer branches represent a higher number of nucleotide substitution, which reflects greater evolutionary divergence. This interpretation is supported by Tamura et al. 2021, who explained that branch length in maximum likelihood trees correspond to the estimated amount of genetic change.
Based on the phylogenetic tree, the taxa that are more closely related are those that branch off from the same relation node and share a common ancestor node shared with the other taxa. These are often referred to as sister taxa. For example, if taxon B and taxon C are located next to each other and connected by a single node, it means they diverge from the same ancestor and are more closely related to each other than to the other taxa in the tree. Their position suggests they have a shorter evolutionary distance between them, both in terms of time and genetic changes. This close relationship could be supported by similarities in their DNA sequences, physical traits, or evolutionary history. In contrast, taxa that branch off earlier in the taxa in the tree or are located farther apart are considered less closely related. By examining how the branches grow and how recently they split, we can determine which taxa are part of the same lineage or clade. So, taxa that are placed as sister groups or that form a small cluster within the tree likely share more genetic similarities and are more recent common ancestry, making them the most closely related in the group.
And the last of this part is 1.4, which taxa is the outgroup and why. The outgroup in this phylogenetic tree is species 2. This is because an outgroup is one or more taxa that are known to have diverged before other taxa in the phylogenetic tree. If an outgroup is too distantly related, it can be unreliable because it may be difficult to reliably align or has become saturated with substitution. Determining the outgroup is important as it helps to root the tree.